Well, thank you for inviting me again. I think this is my third appearance here. I do need to start with correcting the previous speaker on one point. It was a really interesting talk about gene therapy, and I think Dr. Kuhn did a great job. It's amazing research, but Spider-Man did not say, with great power comes great responsibility. It was in the first issue, the first ever issue of a Spider-Man comic book. But it wasn't Spidey, and it wasn't Uncle Ben, which many people believe it was. It was the narrator, who, as Spider-Man is walking away, realizing that if only he had done things differently, his beloved Uncle Ben would still be alive. The narrator says, with great power comes great responsibility. I know this because I just used it in an article I wrote, and I had to come up with the appropriate source and a footnote. But more importantly, with great power does come great responsibility. And CRISPR is great power. I've been around this business, the sort of law and genetics stuff, for about 30 years. And I have seen breakthroughs announced regularly. The Stanford Medical Center's news service puts out a breakthrough at least once a week. And if one actually turns out to be deeply important every couple of years, I think we're doing well. Breakthroughs come and go. True breakthroughs are relatively rare. And I think in my 30 years of watching this, so cheap sequencing is probably the most important, but there's no one moment you can really put on cheap sequencing. CRISPR is a very close second and may actually be the first, because it's not the first time we've been able to edit DNA. I would argue you can go back to Paul Berg and recombinant DNA over 45 years ago for the first editing of DNA in living organisms. But it's the first time it becomes accessible. The analogy I've used that I really like is it's like the Model T Ford. There were cars before the Model T Ford. California probably had, a couple thousand cars before the Model T Ford. They were really expensive. They broke down all the time. You needed to have a full-time mechanic/chauffeur in order to make it work. You had to be rich, and so they were rare. We would not have freeways, smog, suburbs, and the rest of California but for the Model T Ford. Now, people don't drive Model Ts anymore, except as a curiosity, and I don't think people are going to be using CRISPR-Cas9 in 50 years. I don't think they're going to be using CRISPR-Cas9 very much in maybe 5 years. It'll be CRISPR something. Maybe in 50 years it won't even be CRISPR. Bacteria have been around for a long time, and they've invented a lot of very clever ways to do things. But gene editing has become cheap, fast, easy, accurate, in ways that transform the world. It is often the case that the important change is not the first time something's done, but the first time it's done in a way that brings it within widespread use. And that's what CRISPR has done. And I think that's frankly quite revolutionary. Richard and I prepared deeply about how we're going to split up the responsibilities. We had about a 15-second conversation 12 minutes ago about it, but it works out perfectly. I'm not going to talk about people at all. I want to talk about the non-human side of CRISPR, in part because I genuinely think that at least for the next 20 years or so, it's more important than the human side, in part because the Valentine's Day report that he's going to talk about does such a great job of talking about the human issues, and I agree with almost all of it. So humans come after me. I want to talk about the use of CRISPR in non-humans. I first want to tell you about the different kinds of uses I think are going to be made of it, then what the current regulatory structure looks like, and then what the regulatory structure desperately needs to look like, and some of the problems of getting from here to there. Before I do any of that, why do I think the non-human uses are more important? Because we actually care much more about people. We require testing and FDA approval and EMA approval and a variety of things before you can sell a treatment. We have medical malpractice and product liability suits. Human subjects have lawyers. If you're going to mess around with reproduction, you need women who are brave enough to risk their future children this way, as well as physicians and IVF clinics who are willing to risk their reputations, their liability insurance, and their licensure on doing borderline work. You move to non-humans. If you've got a system of using CRISPR in embryos that leads to 3 damaged embryos, 3 damaged babies out of 100, that's horrendously bad and nobody's going to do it. If you've got a system of using CRISPR in mosquitoes that leads to 3 damaged mosquitoes out of 100, people are going to applaud. If it leads to 97 damaged mosquitoes out of 100, people are going to applaud. We don't care about animals that much, let alone about plants and microbes. Except for PETA and a few others, they don't have lawyers. The barriers to moving ahead with non-human uses of CRISPR are very, very low. Both the formal legal barriers and the informal practical barriers. And that's why there could be somebody in a garage, probably not in Palo Alto because even the garages have gotten too expensive, but maybe in Tracy, who is, as we speak, modifying the genes of something in a way that could affect the entire world before we even knew it. So that's why I think it's important. That's not happening with people, I'm pretty sure, but it may well be happening with non-humans. So what sort of non-human uses do I expect? I'll see if I can remember them all. I got 10. The first, the biggest, the one that everybody thinks about is agriculture. And that, I think, deserves to be first. We have had genetically modified crops and livestock basically forever. Our ancestors, the farmers and the herders, were actually excellent geneticists. They're a little slow, but they got things done. Nothing that we eat today looks like it did 10,000 years ago because humans have intervened to do genetic modifications. In the last 30 or 40 years, those modifications have been made more directly. CRISPR makes making those modifications easier and easier. There has been a lot of opposition to GM food. I find it hard to accept. I don't think it's very rational opposition. It's strong opposition in some places, stronger in some countries and cultures than in others. I think ultimately, that's likely to dissipate, particularly as climate change, to which there's also a lot of opposition that I think is irrational to the existence of it. I think climate change is going to force us to use genetic modification for crops because it's a heck of a lot easier to change the genes of corn or wheat than to move the corn and wheat belts 200 miles farther north. It's a lot less disruptive to change the crop than to change where you grow it. So I think even the EU is eventually going to have to fold and accept genetic modification, and it'll be enormously important. One of the interesting aspects of it is so far it's mainly been used as a way to reduce farmers' costs. That's been the selling point. Consumers don't see anything particularly wonderful for themselves in that. To the extent it gets used to improve the product, improve the consumer's experience with the product, I think that'll be an interesting change, or to keep the product available. One of the unsung great examples of genetic modification, of intentional genetic modification of a crop, is papayas in Hawaii, which were going extinct as a result of a disease. Genetically modified papayas now continue to be grown and sold in Hawaii. So things like that, where you're protecting, and there are a whole bunch of crops, including the most popular form of banana, the Cavendish banana, is under serious attack from, forget whether it's a bacterium or a fungus or a virus or something. And people are working on genetically modifying the Cavendish to make it immune to that attack. So I think bioagriculture, agricultural uses of CRISPR are going to be enormous. I think there will also be some interesting uses in terms of biofuels. People have been experimenting with this for a long time. So far, the results have been somewhat disappointing. But the more control you've got over where you're making changes, what changes you're making, and the more time you've got to figure out what changes you need to make, in order to, say, turn algae into great producers of hydrocarbon fuels or hydrocarbon fuel precursors. I think the more you're going to move in that direction. So I think that's one. One that is, I think, often overlooked is bioremediation. There's some interesting things that you can do by using life forms, usually bacteria, that soak up particular pollutants, that differentially concentrate, say, heavy metals, or a problem in the western San Joaquin Valley about 100 miles from here is selenium. High levels of selenium in the ground, which has leached into the water supply and caused the closure of a reservoir when all the ducks were having deformed ducklings, the Kesterson Reservoir about 15 years ago. Figure out genes that help something suck up selenium, plant it in, say, grasses, sow the grasses in the area, harvest the grasses and dispose of them carefully, and over time you decrease the amount of selenium. Maybe you decrease the amount of salt, which is a serious problem in a lot of agricultural areas with salinization of soil. So those are three sort of ag-related kinds of things. Let's talk a little bit about some other uses. Again, I think not looked at that often. Using it in wild organisms. You guys have, I understand, heard about gene drive. So at least to the extent the wild organisms are sexually propagating organisms, gene drive makes all of this more possible, makes it possible to spread it more quickly. A great example, and a wonderful cause to contribute to, by the way, there's a foundation that is doing this research at one of the state universities of New York, is the American chestnut tree. There used to be billions of American chestnut trees. They were the main tree in the eastern North America, and were incredibly important in the ecosystem, especially in the Appalachian Mountains. There are almost no mature American chestnut trees left, because over 100 years ago, a fungus from Asia was imported inadvertently with some Asian chestnut trees, and it killed the American chestnuts. Now, it killed them in an interesting way. This is sort of, everything gets complicated. As a lawyer, one of the things I love about biology is they're very, very similar. They're full of rules that are full of exceptions, that are full of provisos. Everything is fractal, everything gets complicated, and the lower you go, the more levels of complexity you find. This fungus hasn't killed all the chestnut trees, but it kills individual trees before they can become sexually mature. So basically you get prepubescent chestnut trees, and then they die. And when they die, the trunk and the root structure is still alive and puts up another prepubescent chestnut tree, which dies. How long it will take eventually for the root structure and the trunks to give out and die, we don't know yet. So you've got these chestnut trees, but they never make it into their teenage and adult years. People have discovered that oxalic acid, which wheat produces from a single gene, when moved into chestnut trees, will protect them from this blight. And so there's a lot of interest in creating genetically modified chestnut trees, adding a gene that makes oxalic acid, or do you add the gene from wheat, or do you just modify the DNA of the chestnut tree using CRISPR so that it begins to produce oxalic acid on its own? A lot of interest in using that to restore the American chestnut tree. There are black-footed ferrets, an endangered species of little carnivorous mammal, lives around prairie dog colonies. It was on the edge of extinction. They were all captured, put into captive breeding, and they were re-released. Turns out there's a virus that's been wreaking havoc with them. And so now there's an effort to modify, there's thought about modifying the prairie dogs, which is what they eat, and how they get infected with the virus, to make the prairie dogs immune from the virus in order to save the black-footed ferret. CRISPR is one of the methods that's being explored to do that. So managing wildlife is another possibility for CRISPR. The extreme of that, my fifth example, is something I've written on that is, I don't think very important, but a lot of fun: de-extinction. In a way, the chestnut tree is a de-extinction story, but you can imagine taking any organism for which we have a complete DNA sequence, any extinct organism for which we have a DNA sequence, find an existing organism that is relatively similar, and start editing. George Church has been trying this with the mammoth. The mammoth, of course, is the definition of charismatic megafauna. It's what everybody wants to see, although personally I'd go for the giant ground sloth. I think the mammoth is not likely to happen anytime soon, or even the elamoth, even a hybrid between them, because mama Asian elephants are not particularly easy to work with or common. But things like some of the birds that people are working on, the heath hen, which is extinct in New England, was in the mid-Atlantic states, the passenger pigeon, the thylacine. It's a little tricky there because there's not a good host for that. There's not a good sized marsupial similar, but people are working on that. Or the wonderful gastric brooding frog, where the female swallows the eggs, her stomach turns off for 3 weeks, the eggs develop into tadpoles in her stomach, and then she sort of vomits them back out through her mouth. Went extinct again because of a fungus. Lots of amphibians are in big trouble because of fungi that have moved around the world. It's now extinct, but there are relatively good samples. So de-extinction is another potential use for wildlife. I think this is my 6th, changing wildlife to change, and this is kind of about humans, change vectors for human diseases. This has gotten a lot of interest and attention with respect to mosquitoes. In several ways. One is, and all using gene drive, one is to make mosquitoes that have sterile offspring. So you end up greatly reducing the number of mosquitoes in the environment. A company called Oxitec has been doing that and has had field tests in Brazil and Honduras. They've gotten into a buzzsaw of controversy in the Florida Keys, although to the extent Zika shows up in the Florida Keys, that controversy may go away. The more interesting one, in a way, is the work being thought about with respect to malaria. Humans do not get malaria from other humans, except, I suppose, IV drug users or transfusion issues. It's a blood-borne disease. They get it from mosquitoes. The Anopheles genus of mosquito carries malaria, carries the human form of the malarial plasmodium. What if you make the Anopheles mosquito immune to infection with the malarial plasmodium? No infected mosquitoes, no infected humans. That's about 600,000 human deaths a year you could stop, in theory, if you could change the mosquito so it can't carry malaria. And again, people in Switzerland or Germany or the United States may say, oh, that's terrible and unnatural. Mothers who have lost their infants in Africa or South America or Southeast Asia may have a different view about what's appropriate and what's not. The other one that's really interesting, and Kevin Esvelt, I suppose one of, if not the father of gene drive, has been working on this. He's a really interesting guy at MIT, very into public communication and transparency in a way that I think is worth emulating, certainly worth learning about. Kevin's working on Lyme disease. As a Californian, I barely know what Lyme disease is. But in the Northeast, particularly in New England, Lyme disease is a huge issue, maybe more of a panic than an actual health issue, but certainly some health issue and a lot of panic as well. And it's particularly common on these 2 rich little islands of Massachusetts, Nantucket and Martha's Vineyard. And so Kevin, I think he's working at the Vineyard. I can't remember which island he's working on. His goal eventually is to genetically modify one of the steps in the infectious cascade and the infectious procession with respect to Lyme disease. He's working on a mouse. So ultimately they're passed to humans on ticks, but the ticks get it from the mice. So he's got the white-footed mouse, which is the native mouse species on the island, and he wants to make those mice immune to Lyme disease. And like the malaria story, if you make the mouse immune to Lyme disease, the ticks don't get Lyme disease, so the humans don't get Lyme disease. It's interesting because the higher income places tend to be a little more not in my backyard, but Lyme disease is apparently quite worrisome to people who live in Nantucket or summer in Nantucket.
It's their summer backyard.
Yeah, it's their summer backyard, right. And so he's hopeful that in 10 years or so he's going to get a chance to try it. So that's the 6th example. Let me move to some other uses, domestic uses. People tend to think about good, important, valuable things, but a lot of stuff gets done frivolously or semi-frivolously. You know, the biggest use of genetic testing in America is for ancestry testing, which is kind of fun, but for most of us isn't very important, which is a good thing because it isn't all that accurate either. Paternity testing, yes. Broader ancestry testing. My ancestry is mainly European. I didn't actually need to pay $99 to discover that. Although if it turned out otherwise, it would have been very interesting. But consider first pets. How many of you have or have had a dog? Okay. 10,000, 50,000 years ago, those were wolves. Any of you have like little dogs, big rats, chihuahuas, Shih Tzu? Oh, come on, some of you do or have, yeah. And any of you have really big dogs, St. Bernards, German Shepherds, Great Danes, Irish Wolfhounds? Now, they were all wolves. We turned them into this. And actually, we did most of the transformation just in the last 200 years. A few of the breeds go back farther, but most of the currently viewed classic breeds are only a few hundred years old. We did that genetically. Well, people are interested in, say, hypoallergenic dogs and cats. You can do that through breeding. You can probably also do that through CRISPR. You don't have to make Labradoodles or whatever. You can make any dog you want or any cat you want to be hypoallergenic. So I think we'll use it for pets. Any of you gardeners? Anybody grow roses? Nobody grows roses? A little bit. Anybody ever see?
With difficulty.
Yeah. You need to move here. And this is a great climate for roses. At least Palo Alto is. Berkeley probably too wet. Anybody ever grown a blue rose? No one has made a blue rose. There's a Taiwanese group that's made sort of a lavenderish rose, but it's not a blue rose. I'm not sure why somebody hasn't done this already. I'm sure, like everything in biology, it will turn out to be more complicated than it seems. But take a white rose, add blue genes from hydrangea or other really deep, powerfully blue plants, and make a blue rose. Gardeners have been trying to breed for blue roses for 200 years. CRISPR should make that easy. People will use it for these kinds of things. My co-author, there's a paper that I did with Alta Chara, about whom you will be hearing more in a moment, in the American Journal on Bioethics, January of 16, something like that, called CRISPR Critters and CRISPR Cracks. And Alta pointed out a use I hadn't thought of. There are a whole bunch of food crops, of particularly fruits and vegetables of varieties that don't exist anymore, that look beautiful in the old paintings. Well, if anybody has DNA remnants from, say, 16th century plums, sequence them, make variations in the modern ones, and you can recreate old crops. So I think there'll be a fair amount of that. There will also be people who do this just to make an exhibition. You know, put it in the circus. I think there will be a unicorn. Now, this is a little farther away because I don't know of any beasts that currently have just one horn coming out of their forehead. The narwhal, whose horns were sold to European monarchs as unicorn horns. It's actually a tooth coming out of their jaw. But figure out how to make a single horn from a forehead, add it to a horse. I predict, and this is a fairly serious prediction, that within 20 years, some Silicon Valley billionaire will have a unicorn made for his 12-year-old daughter. And I'm pretty sure it'll be a him, and it'll be for a daughter. And the unicorn will be nice and white and beautiful, etc. People will do it. They'll make dragons. Now, they're not going to repeal the laws of physics. The dragons won't fly. And I used to say they wouldn't breathe fire, but then I learned somebody who's been working on modifying a lizard, so it actually does kind of breathe fire, so that it exhales volatile fumes that could be set on fire. But you know, you can't make this stuff up. That's actually real. People will do this because it'll be cool, and maybe they can make a lot of money showing off their dragons or whatever. Ninth category, artists. There's a fair amount of concern in our culture about the mad scientist. With one or two possible exceptions, I don't think I've ever met a mad scientist. There's one at Harvard Medical School who might qualify, but for the most part, scientists are relatively constrained. You know, not only are they people who've done well by following the rules that I think are selected to be relatively rule-abiding, but they need jobs and tenure and grants and Nobel Prizes and stuff like that. And to be really outrageous probably doesn't help advance any of those. A few people can get away with it, although even Jim Watson got more outrageous after he got his Nobel Prize rather than before. But for the most part, scientists are too constrained by the system to be outrageous. But there's a group of people who like being outrageous, at least some of them, artists. And there are bio-artists in existence now, genetic artists doing and trying to do really weird things. Sometime Google Adam Zarefsky, Z-A-R-E-F-S-K-Y. He's out there, but he's not alone. Genetic art, making new creations, making living creations as art, and the more outrageous the better as far as the artist is concerned. That, I think, is actually what's going to make things that make the public queasy much more than the scientists are. Last category, 10 out of 10. Biowar. Human pathogens. So humans, again, to some extent, but not in a way you're going to talk about. Just a report recently, Canadian group made horsepox. Horsepox has been extinct. They knew the sequence of horsepox. They recreated from the ground up horsepox. There are lots of pox viruses out there. There's mousepox and horsepox and monkeypox, and by the way, there's this thing called smallpox. We hope that smallpox currently exists. Lives is always a little tricky for viruses. I think I'm of the position that they're not alive, although we can kill them. But we think it exists only at CDC in Atlanta and in a lab in Russia, I think in Moscow. But we don't know that for sure. And how hard is it going to be for somebody? The sequence of smallpox is known. The sequence of monkeypox is known. You take one, add CRISPR, add a garage, add about $5,000 of equipment bought over eBay, and you could do this. Probably somebody with an undergraduate degree in biology who is smart and tenacious could do this. Graduate students would be better. Postdocs, yeah, who knows, but certainly PIs would be terrible at it. But you could do this. And by the way, it doesn't have to be just the lone wolf crazy. And there are lone wolf crazies out there. Some of you, it's long enough ago that you may not recall, but there was a guy named Ted Kaczynski, the Unabomber. And one of his issues was genetics. He blew off the hand of an eminent geneticist with a bomb because he thought genetics was doing terrible things. A Kaczynski who decides to recreate smallpox would be really terrifying. But you can also look at organizations. ISIS held Mosul for, I'm happy to be able to say that in the past tense, although Mosul itself may now be in the past tense. I think it was pretty heavily damaged. But they held it for 2 years or so. Home of the second best medical school in Iraq, a modern medical school. Supposedly, and this came out just about the time of the summit on human gene editing in Washington, they executed the head of the physics department at the University of Mosul for refusing to help them make a biological weapon. So terrorist organizations, let alone rogue states, as well as individual lone wolves, could use CRISPR for biological warfare purposes. The good news, to the extent I can say there's any good news, is that biowar is tricky. It's not just having the pathogen. You've got to have a good delivery system, and you've got to have a way of protecting your own people. But the pathogen is an important first step. And CRISPR makes getting those pathogens, either taking an accessible organism and modifying it, or in the not too distant future, building from the ground up and making modifications, makes it much more possible. And that's something that should keep us awake at night. And that, I think, has the potential for happening a heck of a lot sooner than super babies through CRISPR embryo editing. So I went on for a while there, but I do think that people tend to have tunnel vision about the uses of CRISPR in the non-human world and think mainly about crops, and then if they've heard about it, maybe a little bit about the mosquito stuff. There's a lot of potential there. What is the current regulatory structure? That's a great question. The answer is, the honest answer is, who knows? It varies from country to country. In some countries, GMs are very, very tightly restricted or banned, but the definition of genetically modified is unclear. Other countries allow them under different regulatory schemes. Again, the definitional issues make trouble. The United States, since the 1980s, mid-1980s, has operated under something called the Coordinated Framework. When GM foods first were introduced, the Reagan administration wanted to have a pathway for them to come into being, but it recognized, I think accurately, that it wasn't going to get anything new through Congress that it liked. So it said, well, we'll look at the existing authorities for FDA, for USDA, US Department of Agriculture, and for the Environmental Protection Agency, and look to see what we can do with their various statutes, and we'll assign these responsibilities to the various departments. And it's worked, from some views, reasonably well. I think the people who are finally getting approval to sell genetically modified salmon after 20 years would say it hasn't worked so well for them. But for a lot of crops, it's worked pretty well, reasonably well. The problem is the statutes had nothing to do with genetic modification. Stretching them to the extreme kind of gets most of the stuff, but it still leaves gaps. So for example, somebody came up using CRISPR with a mushroom, the sort of cheap, cheesy little white button mushrooms that you see everywhere, a mushroom that does not brown when you cut it. So normally you cut mushrooms, and after a while you don't have nice white mushroom slices, you've got icky brown mushroom slices. These may still be icky, but they're not brown. Took out a gene, deleted a gene responsible for the oxidation that helped lead to the oxidation process that turned them brown. Went to the USDA to see if it needed to get approval. The USDA said, we don't have jurisdiction over this. We only have jurisdiction over crops where a gene was moved from one species into another. In this, no gene was moved, and the CRISPR construct that was involved, even if we expand our thought of gene to include the guide RNA, is gone. It's no longer in this mushroom. We don't have any jurisdiction over it. Who has jurisdiction over it? Nobody. Unless, well, no, nobody. Unless maybe you thought about browning as a plant disease, in which case another part of USDA might get involved, but it didn't. Now, the Obama administration noticed this, and the Office of Science, Technology, and Policy, which I'm tempted to say the late lamented Office of Science, Technology, and Policy, because it's gone from 135 staffers to 35, with 0 in their science division, literally 0 people in the science division of the Office of Science, Technology, and Policy, which exists to advise the White House about science. Draw your own conclusions. It said, we need to think about this, and it asked FDA, USDA, EPA to think about it, and just before January 20th, 2017, a day that will live in infamy, the FDA and the USDA came out with some proposals about how they might regulate it. Similarly, the National Academy of Sciences came out with a report on gene drive in 2016 and a report on a larger biotech framework in 2017. I don't think any of those are good enough. And the ones that came out from FDA and USDA, some were just requests for thoughts, requests for comments. That was USDA's. FDA's were relatively narrow, and we don't know what the new administration is going to do with them. They're proposals that either need to be finished or not. The National Academy reports were very good discussions of the topics and the issues. But as sometimes happens in National Academy reports, I think they shied away from getting too detailed in terms of their policy prescriptions. There's a concern sometimes in the Academy about not wanting to tell Congress what to do. So I don't think they're all that, they're helpful background, they're not helpful going forward. What should we do? Roman numeral 3, the final Roman numeral of my talk, what should we do? If I was put in charge of creating a system for regulating uses of CRISPR and other genome editing in non-humans, here are the basics of it. First, some sort of registration and labeling scheme, so that anybody who is going to use genome editing in a living organism or virus would have to be registered with presumably the federal government, could be somebody else, but has to be registered with somebody. This is not going to be a big problem for university researchers or corporate researchers, Monsanto or Bayer or think tanks, Salk and Sabin. You know, they're going to be able to do this. They got systems for doing this. This is intended mainly to catch the small people, the lone wolves, the ones who are doing it because it's fun. And by the way, there is a whole do-it-yourself CRISPR movement as part of the do-it-yourself bio movement. Somebody, I think it was on, I can't remember whether it was on Kickstarter or Indiegogo, but one of the crowdfunding sites funded do-it-yourself CRISPR kits, and for 99 bucks, you can buy some CRISPR that will allow you to make modifications to bacteria that they send you with the kit. So there's a lot of interest in this. I say first, make sure those people are registered, and as part of the registration, make sure that anything that they transform, they label. Now, this is not like the label that comes on pillows that say, do not tear this, against law to tear off this label. And actually, this is one of those areas where DNA is so interesting and cool. How many of you saw the thing yesterday, I think, about CRISPR being used to put in a GIF about a running horse? And just as an information storage technology, well, you give everybody a code number or a base pair code, and say, this is yours, and you have to put this into every product you make. Craig Venter, of course, already did that, but I think it was Craig's name that he put into the bacteria that he used. But you could have, and you think about it, 4 base pairs, a 10-letter string is 4 to the 10th, which is 2 to the 20th, which is 1 million already. You add a couple of more, you get plenty of things. So you say, you have to register, and anything you make has to have your 15 base pair code in it. Second, you have to tell somebody what organisms you are planning to modify, and at least in general, what kind of modifications you're making. I'm not, at this point, convinced that you should have to get approval for those, but you should have to record it and make sure that somebody knows it. So in case a genetically modified E. coli gets loose, and you haven't done your labeling, there's some sense of which labs and what areas were working with E. coli in which sorts of ways. So some sort of notification. I would add to that, the notification could lead to a ban in some uses. If you notify somebody saying, I'm planning to modify mousepox to make it more like smallpox, that shouldn't be allowed. So some sort of review of what you're saying to look for really dangerous stuff, particularly if it's really dangerous stuff that you don't seem equipped to deal with. You don't have a class 4 lab in terms of biosecurity, let's say, and you want to play around with extremely pathogenic human viruses, probably don't allow that. Third thing, release, environmental release. Part of this is avoid inadvertent release. So these are biosecurity measures to keep things in the lab from sneaking out of the lab. And we've got a lot of experience with this. This was actually the main upshot of the Asilomar meetings about recombinant DNA. They went a little bit overboard, but as time went on, the requirements got cut back a little bit. So ways of making sure saying you recognize you're not allowed to let these mammoths run wild. You've got to keep them in your lab or in your facility. And here's what you have to do to worry about microbes or fungi or viruses, et cetera, getting loose. And then when you want to release into the environment, whether it's commercially or not, some sort of approval process that requires somebody impartial and expert to assess the safety of what you're doing. I think a risk-based thing. If it's a blue rose, that seems to be relatively low risk. Just like the mushrooms, I got no really worry about the fact that the USDA isn't approving these non-browning mushrooms. I don't think that's likely to be a big deal, but it's good to have somebody take a look and see if there are any possible concerns. Plus, it's good for establishing or trying to establish some public trust that things that are being put out there have actually been looked at. So some sort of review, and that review, for some things will be pretty straightforward. For others, it may say, okay, first you need to do a trial, either in an enclosed space, sort of an ecological trial, or you need to do it on an island or do something else and monitor it and see what the ecological ramifications are. What you're mainly worried about with the releases, I think, are environmental and ecological ramifications. A little bit with maybe allergens or disease vectors that you've inadvertently created or so on. But mainly, you don't want to be the person who releases kudzu into the southeast US, an invasive plant from Asia that somebody thought would be useful and has now taken over most of Alabama, which may or may not be a loss. But or other invasive species. The story I like best here is, you guys know what starlings are, little black birds? There are now hundreds of millions of them in North America. They are not native to North America. They are loud, they kick out other native species, they're dirty, they're noisy, they flock in huge flocks, they're a pain in the butt. They exist in North America because in the 1880s, a Shakespeare enthusiast decided it would be nice if every bird mentioned by the bard was in the United States. So he brought some breeding pairs of starlings to Central Park. Started with like 12 breeding pairs of starlings, 120 years ago, 130 years ago. So invasive species are a real concern. So that's the main thing I think the approval process should look at, the ecological and environmental effects, but there are other things it could look at as well. Last step, plans for cleaning up the mess. What happens if things go wrong? Have some plans for it. Some of those plans might be sophisticated, like kill switches, ways of nicely terminating the creatures you left out. Some might be easy ways to identify them. So green fluorescent protein, so when you catch one, you can see quickly in an expressed area. You catch one, you can see quickly whether or not it's the GM strain or not. Some of them with gene drive. Kevin Esfeld has had some ideas about ways to have gene drive peter out after a few generations. Yeah, a self-limiting gene drive.
Daisy drives.
Daisy drives, yeah, that's what he calls them. And then sort of brainless, mind-dead stuff that's useful, like insurance requirements or bonding requirements, so that if what you let out makes trouble, you can pay for at least some of the damage. So that's my sketch of what kind of regulatory system I think we should have. Here are 3 reasons why we're unlikely to get it. First, even if by some, well, first, getting the lone wolves to register is going to be hard, especially when it's easy to get the CRISPR stuff, like through do-it-yourself CRISPR kits. We have kind of an analog to this with drones. The Federal Aviation Administration wants everybody to register their drone. So you buy a drone at a toy store for $50. It comes with a piece of paper you're supposed to send in to the FAA. And kind of to my surprise, hundreds of thousands of people have sent them in, but millions of people haven't. How do you figure out who's actually doing this stuff? Especially if they're doing it sort of cleverly, buying stuff through eBay, buying it used, getting it from friends, getting it from their work or their school. How do you know who's actually getting it, and how do you get them signed up? Second.
So just
yeah.
a thought on that. If there are penalties for not obeying these regulations, then you can potentially get one of these rogue garage scientists when he's caught, and there'll be a penalty.
Right.
Sort of a poor analogy is getting Al Capone for tax evasion.
Right. So there could be deterrent effects. Having punishment afterwards gets more people interested in obeying the law in advance. As the legal system believes that works some, the legal system knows that doesn't work perfectly. I did not obey the speed limit. It was an unusual day. I got from Stanford to here with the chance of, with the opportunity to break the speed limit, which I happily took. Not by very much. And murder has been illegal for a long, long time, but it still happens. So yeah, the deterrent effect of penalties can play a role, but it's an issue. Second problem, and I think, to me, this is a bigger problem. I've set out what seems to me a reasonable, appropriate approach. I hope to publish it and then get other people to talk about it and think about it. Sort of approach that I kind of wish the academies had talked in more detail about, implementing some of the general positions, not your report, but the non-human academies reports. I don't have, beyond the faintest glimmer of hope, that Congress would pass something like this. First, right now, our Congress will not pass 2 + 2 = 4, because it'll be a partisan issue. Fake arithmetic, you know. We're in a stage where getting anything through is difficult. I think getting anything through that's sensible about genetic modification is going to be really difficult. And I say that I have my own partisan views, but regardless of which party is in power in Congress, I think it's going to be a real problem to get good regulation passed through Congress. And yet I think it has to go through Congress because the existing laws don't give you enough scope. They weren't written with anything like this in mind. So I think that's the second barrier. And the third barrier is, let's say that somehow miraculously, Congress appoints me the czar of all GM, and I'm just allowed to do this. Well, that's one country with 5% of the world's population. What do you do with every place else? And we know that there are great cultural differences in countries in terms of how willing they are to do these things and not do these things. And by the way, the other issue is, if most countries ban it, it becomes then a marketing opportunity for somebody else. There are tax havens and banking havens and reproductive havens so that people can avoid restrictions in their country by going to someplace else. The idea of getting any sort of consistent, rational, worldwide, with teeth, enforceable system for regulating this seems to me a fantasy. And yet, it's crucial. If you change mosquitoes in, say, the Central African Republic, there's no reason to think they're going to stay in the Central African Republic. They're going to spread, and they may spread over the entire world. This is one of those technologies, kind of like geoengineering, that somebody in one spot can do that can affect, I said, which may be right, can affect the entire world. And getting some universal regulation for it, I think, is going to be politically really hard because countries will have different views. And legally even harder still. So, having said all that, I'm still in favor of talking about it, writing about it, thinking about it, pushing for it. We may not get perfect, but you can't let perfect be the enemy of good. And what we have now is effectively no real regulation at all, or very limited real regulation at all. I'm a lawyer by training, and hence arguably cynical. I prefer to think of it as skeptical. I think what it will take is a disaster. Something really bad happens, and when something bad happens, legislatures have a tendency to look around for something they can say will fix it. It's like after 9/11, the USA Patriot Act, the world's largest and clumsiest acronym, by the way, it stands for something, had almost nothing to do with hijackers taking over airplanes and running them into towers. It was a bunch of ideas that people had had to tighten security over the last 10 or 15 years that had been floating around, and then the political impetus came for it. The FDA exists in its current form with regulation of efficacy because of thalidomide, which was a safety problem, not an efficacy problem, but led to the political will to change. So I welcome your questions, your thoughts, particularly your suggestions about what a regulatory scheme, whether the scheme I suggest or some other would be good, where it's too strong, where it's too binding, where it's too weak, and thanks for your attention. See.
Just to get this straight, so with your example about the guy with the mushroom,
Yeah.
the non-brownie mushroom, so where is that right now? Is he allowed to?
He's allowed to sell it. Yep.
I'm not absolutely sure that's true. I think the USDA said he can grow it. But to sell it as a food, I think FDA also has to give approval. That's what we heard about yesterday from a guy from Calyx. That,
Well,
so it's the Food
I'm
and
not
Drug Administration.
Yeah, but the FDA's approval over foods is generally limited to food additives. Now, there are a lot of your foods will say approved by the Pennsylvania Department of Agriculture. There are some state regs. The FDA, well, I'll be interested.
Calyx has.
And they should know,
Yeah,
right?
they have plants in the field.
Yeah.
You showed us acres and acres of soybean and potatoes, but they can't put them on the market. They have USDA, it's not approval, it's the same as the mushroom.
Permission, yeah.
Not within their purview.
So I'll have to...
But in order to actually put them on the market, they have to get FDA approval.
I have to look at that. I'm not sure that's right, but the fact that a company doing it thinks it's right is some evidence, some pretty good evidence.
My only source.
Yeah, and livestock, meat is treated differently than plants by FDA.
Right.
So, but FDA, USDA, and the other agencies involved in the combined framework have allowed a variety of things in, including all the Roundup Ready crops, the Beta Thuringiensis crops, the papayas that are virus safe, etc.
But there's absolutely no rules against him making the mushroom, like designing that kind of mushroom. It might only be.
There may or may not be rules on selling. The USDA position is it's got no jurisdiction unless he's moved a gene from one species into another. Yeah.
What's the situation in Australia? Is there a similar criterion for allowing production?
Yeah, I'm not sure. I'm really interested to find out now. I wonder if it is similar. 'Cause
Yeah.
We haven't had any. There's been no news of anyone genetically modifying food that hasn't been cleared. So
The definitional issues are getting interesting because when this came up in the 80s, it was things like moving genes from deep water fish into tomatoes to make them strawberries to make them freeze resistant. And so it was the transgenic side. There is some of the pro-GMO people have this hope that, well, it's the fact that you're moving it from one species to another that freaks people out and allows Greenpeace and other opponents to get political support. I think that's probably a vain hope. I think that's probably wishful thinking.
We had a meeting in Europe and we heard from a number of the European regulators,
Yeah,
including the discussion of GMO foods came up.
yeah.
There is a move among some people in Europe that if there's nothing left that's
Right.
foreign, it's not genetically modified. And if that could be pursued, that might be a way around that. It's basically a linguistic block.
Right, although I think that's probably more the case with the regulators than certainly it is with the activists.
Yes.
Greenpeace is not going to accept
No,
this.
but
And
they're
whether the
not the
political,
majority.
but where the political opposition is coming from, I think the regulators probably think what they're doing already is kind of crazy, and so are looking for ways around it. Whether they can make it stick in their political systems will be an interesting question. Yeah.
So I was hoping you could comment a little bit on the role of the judiciary in this, because obviously we need new laws that will encompass this stuff, but while we're waiting for that, we have the judicial system trying to interpret old laws that don't encompass modern genetic ideas.
Yeah.
And we're sort of stuck iterating on what's in the language.
So there are a couple of aspects to that. The American judiciary, when it's dealing with regulations, tends to give a lot of deference to the regulatory agency. There's a whole branch of law called administrative law based on that relationship. And so if FDA or USDA says this is how we interpret the regulation, the courts are supposed to give a lot of weight to their interpretation of the regulation, which makes me think that the regulatory agencies are the key issue on the regulation side, not so much the courts. Although there are thousands of judges in the US, and you can bring the suit, you may be able to bring the suit anywhere. So finding a particularly anti or pro-judge, forum shopping is what we call it, could lead to an early decision one way or the other, but the appellate system, I think, is likely to generally though not always, adhere to what the regulators think. There is another whole area, though, of common law tort and product liability. So it's like if today, when I was breaking the speed limit, I rear-ended somebody on Highway 280, when it came, as it inevitably did, to a halt, actually it was 101 that came to the halt, I could be sued. You put out something that causes damage, you could be sued. And I think there are genuine interesting questions about whether a GM modification would be viewed as something where you're only liable if you did something negligently, you sort of screwed up, you made a mistake, whether you'd be liable, or whether you'd be liable for anything you did, because there's this class of tort situations called ultra-hazardous activities. If you keep wild animals in your backyard, even if you've got a completely non-negligent way of keeping them safe, if one gets out and hurts somebody, you're liable regardless of whether you were negligent or not. So sort of strict liability versus negligent liability could be an interesting issue. And that's one area where the courts could make a difference. And particularly, you could end up in state courts where the politics are a little crazier and less predictable than at the federal court level. Other questions? Yeah. So 3 in a row. What are the odds? It's statistically significant. Could be ascertainment bias.
So bring up the states. Could the states lead? Well, I'm thinking of.
Yes, no, and maybe.
Well, what happened with exhaust emissions?
Yeah.
I'm trying to use that.
Yeah, so the state exhaust emissions is a really good example. California led, the Fed said, no, you can't, but we'll let you have special rules as a matter of federal law. So the general rule in the US is federal law trumps all state law. I hate the fact that I can't use that verb anymore without being self-conscious about it. Federal law overrules state law, is dominant over. If there's a federal law that's relevant. And I think to the extent that the FDA, USDA, et cetera, claim that they have jurisdiction, that probably would preempt, but some of it will depend on the language of the statute. So some statutes specifically allow states to go farther. There's a funky case out of Hawaii. The county of Kauai wanted to ban GMs, and a federal court said, no, you can't, because you, the county, don't have the power. The state may or may not have the power. The feds may or may not have the power, but the county doesn't. So this could keep lawyers busy for a long time. However, I think you make a really good point. Even if they may not have legal force, states could, even if it's not clear to what extent they have legal force, by starting to regulate, they can begin to establish norms and standards in a sense of what where the conversation should be. We also have seen in California a couple of counties try to ban GMs, although I think those were initiatives that have all failed so far. Other questions from anybody who's not sitting right there? That's a real mutational hotspot. Okay, well, thanks very much for your time and attention.